Good morning. This is Michael Dong speaking on episode number 18 on chromatography data system or CDS. We will discuss perspectives, operating principles, and show some case studies. We welcome LCGC International as our channel collaborator. Let's begin with the agenda, consisting of three different parts. The first one is perspective. We have an introduction, glossary, requirement, characteristic, and some historical perspective. Second is the principle of CDS and some example, starting from sample testing workflow process and using illustrative example from Waters and Power, showing the data flow scheme, integration, result, spectral display, and databases. Finally, several case studies from Agilent and Thermo Scientific, and also a listing of HPLC system and CDS. A special thanks to Bob Masseries, who is a former sales director of Waters for several decades, who actually wrote a white paper in 2019 with myself, which is contributed to the technical content of this video. Here's an introduction to CDS. First, what is CDS? CDS stands for Chromatography Data System and is used to acquire, integrate, quantitate, and report data produced by a chromatographic system, such as HPLC, GC, or IC. For laboratory performing regulated testing for QC, for quality control, in both pharmaceutical development or manufacturing, the CDS is typically a validated client-server networking system designed to provide data security and integrity. Here's a listing of current version from the four major manufacturer of HPLC, also happen to be the major manufacturer for CDS. These are Waters and Power 3.8.1, Agilent OpenLab 2.8, Thermo Chromeleon 7.4, and Shimatsu Lab Solution 5.3. Some marketing landscape provided by Glenn Cloutier-Met in 2019 show you the market size shown here. Some of the major manufacturer as mentioned previously. I want to say that many CDS software provide control and data handling for both the UV detection, other detectors, and also low resolution mass spec. For many years, Waters and Power dominated the regulated market for QC for 2 decades prior to 2010. However, after the date, others are catching up, primarily Chromeleon and also OpenLab. There's smaller vendors which cater to smaller installations and also their own brands of chromatographic or purification instruments. Some examples are Clarity, a product called Data Apex, Chrome Perfect, ChromeNav 2.0 by Jasco, and Chimera Total Chrome by PerkinElmer. Okay, this is the glossary of terminology. Would not comment too much. 21 CFR, Part 11 CFR stands for Code of Federal Regulation. That defines the criteria for electronic record and signatures. And make sure they are traceable, trustworthy, reliable, and equivalent to paper record. Calibration is the process to quantitate and analyze using a calibration sample or reference standard. I mentioned that most pharmaceutical companies use the client-server network because of compliance and also for data archival and other terminologies. In addition, we talk about terminologies such as instrumental control, the integration process, metadata, which is a set of data that describe and give information to other data. It's not actual raw data, it's meta, something like that. This raw data, where which are derived from the chromatographic instrument from the detector output with a time axis. And these are transformed through the integration process into a peak table, eventually to a report. Most CDS use a relational database, which we will comment further. More acronyms for the future of the laboratory shown here, and here is a list of acronyms such as Electronic Lab Notebook. LIM stands for Laboratory Management System, Information Management System, and LOTF stands for Lab of the Future, and also CFA. And some of the regulatory terms such as GMP, GLP, system suitability testing, and also different instrumental acronyms. This is a listing of requirement and functionality expected in the CDS. Functionality such as data acquisition, data processing through a process such as peak integration, identification, calibration through a reference standard, and report generation, and data archival. Most CDS is also capable of instrumentation control and provide a single point instrument control for all the instrument modules, including the pumps, autosampler, column oven, and detector. For regulatory compliance, one expects the CDS provide data security, traceability, and data integrity in compliance to the GMP regulation and also to the regulation pertaining to electronic data, such as 21 CFR Part 11. Here's a listing of desirable characteristics of a CDS. I would not comment too much. This is a listing for your information, such as multi-vendor connectivity, ability to control, take data from multiple instruments, multiple lab, multiple detector, and also supporting multiple language connectivity. And those are some of the finer detail controlled chromatographic instrument process display data. System should be scalable from single workstation to a global multi-site network. Also, networking and operating system compatible to different operating system, including Windows unit, and also providing remote access from a remote location such as home, using a browser such as the Microsoft Edge or Google Chrome, and including data access using a mobile device such as smartphones and tablet. Another requirement such as data security, flexibility, and archival, using database relational database such as Oracle or SQL Server. Some historical perspective from the 1980s. Before that, most data are handled by a chart recorder by using a manual measuring of the peak height or peak area, using a ruler, by multiplying the height of the peak and also the bay width. In the 1980s, with the advent of electronic integrator, things become much more sophisticated. Shown here is a electronic integrator capable of, with a thermal printer, with a disk for data storage. One can do internal and external calibration, and also can do some basic programming to customize calculation. Unfortunately, the electronic integrators time frame actually shortened by the rapid introduction of PC workstation in the 1980. First debut is by Nelson Analytical, which got acquired by PerkinElmer in the 1980, which quickly migrate into a data handling network such as client-server network. This figure illustrates or highlights the process of a pharmaceutical analytical testing workflow, which operate under an external regulations framework and also internal quality system, right? So external regulation, some of these shown here, GMP, GLP, and regulation for electronic data and electronic signature, and also internal quality system, SOP, the need for specification, method validation, things like these in the and on top of these analytical workflow. We divide the workflow into three different segments. First is sample and also reference standard preparation. Include sampling, sample mobile phase preparation, also documentation through electronic notebook or lab notebook documentation. And then transporting after sample or prepare, transport all the vial right to the analytical chromatographic system to perform analytical testing. Now, some of these functions are actually performed on the CDS, such as instrumental setup using a method, set up a sequence of sample and calibration vials, start the sequence and start the data acquisition. Typically, one would look at real data monitoring for the first few samples, make sure everything's working properly. And then when everything's collected, we would do a batch data processing in general, meaning integration, calibration, checking system suitability, testing result, and definitely doing a batch reprocessing to generate reports. Finally, these data are transferred through another process to generate a CFA, this certificate of analysis. First of all, data is first archived, then went through a QC data review of the technical data by the QC manager and sign off. A lot of time is exported to the LIMS, the data exported to a LIMS system for either CFA generation automatically or in a hybrid format. The CFA is reviewed by the QA, which actually performed the batch release. Once again, this workflow is first documented in this white paper, written mostly by Bob Masseries, a CDS Perspective, Principle, and Trend in LCGC North America magazine in 2019. So the viewers of this video is actually refer to this white paper, CDS white paper, for more detailed description of these processes. Case study number 1 is Watson Power version 3. To give you a little more detail on the terminology and the different type of CDS method type. A quick reminder that the Empower actually was first debuted in the 1990s. So a lot of these method sets take quite a while to get used to it. Typically, one need several days and weeks of training and to make sure the user lab analyst is comfortable with these processes. So after data acquisition, we generate a set of raw data, which is archived, and then go through a processing method or method set, which incorporate the sample and standard information to generate a result or result table. And then the results are actually transformed into a report using a report method or report method set, which format the report appropriately for different assays and also incorporate any type of custom calculation to generate a report. the report is actually what is eventually archived into the CFA. Here's an example of the screenshot from Watson Power showing real-time monitoring of both the UV data and the mass spec data, and also instrumental and sample status, right? So most CDS can allow for real-time monitoring and also for remote access, right? So shown here is the UV channel at a specific wavelength, showing the UV chromatogram. And also this is the actual mass spec tick channel, which is the total ion data shown here, which is look kind of similar to the UV data, and the actual mass spectrum occurring at this point. The lower segment contain the instrument and sample status, right? So you have the instrument data, and also green light meaning that everything is okay. Red light means there's some problem with this module, right? So each module being monitored and with a signal light indication. And also you have the two-channel monitoring and also information on the particular sample being injected. Here we demonstrate or illustrate the integration algorithm called the traditional integration algorithm. Typically, in a traditional format, the way the CDS look at a peak and transform raw data into a peak data is looking at the baseline. When there's no peak emerging from the detector or flow cell, you have a flat baseline. And once you have a liftoff of the data, the CDS suspect that there's a peak emerging, and the user actually would set up a area threshold. Once the threshold is exceeded, indication to the CDS that a peak is emerging. And then when you keep tracking the data, coming to the apex of data, the value, the peak height of the data actually would go up and go down, right? So once it go down, it indicates to the CDS that actually we have the apex of the peak, which is the retention time of the emergent peak. Watson Power allowed one to use two types of integration algorithm. One is called the traditional integration algorithm, which I said before. Now, for complex chromatogram, I would advise a user to use something called apex track, which is a much more sophisticated data. Instead of using the raw data, use the second derivative of the raw data, which allow a much more rational or scientifically feasible way to integrate these type of merging peaks, and shown here in there. And it can do it in a very automated fashion without a manual integration or user intervention. So definitely one should consider the use of apex track as a default for more complex chromatograms. Shown here is some common integration and identification error. So if you have a noisy, if you have a peak, you can actually show the baseline of how the CDS look at the baseline. And clearly this one is not correct. This baseline is correctly drawn. With the noisy peak, sometimes the CDS can indicate multiple peak, which is not correct. And that means the area threshold has to be lower. And here is misidentification. Instead of ABC, there's some retention shift, and A and B and C is misidentified. Here's another example of Empower actually illustrated the capability of a modern CDS to actually integrate both the UV data and mass spec data. Now, historically, that was very difficult to do. And And if you actually purchased a mass spec, particular single quad mass spec from the same vendor for CDS from the same instrument HPLC vendor and the CDS vendor, one have the option of really integrating in a more automated fashion. Now, in Empower 3, there's a magic button called the mass analysis button. If you purchase a single quad mass spec such as the QDA, once you press this button, some really nice automated annotation happen. For instance, it would automatically annotate from the QDA data for the base peak or the most prominent peak, such as M+1 peak from your drugged APIs automatically, which is very, very powerful. And in the peak table, you can see that besides some of the common items such as retention time, area percentage, retention time ratio, you also integrate the data from your DAD or PDA, such as lambda max, and also shown here is the base peak, the most prominent mass spec peak, typically the M+1 peak or M+1/2 for dimer peak. Also, other important table items are USP resolution, USP tailing, and signal to noise ratio. All these can be extracted into automatically in Empower peak table. So very nice time-saving CDS by integrating both UV and mass spec data. Most modern CDS actually use a database technology. Instead of using a flat file format, a relational database is used to allow you to have a bunch of benefits, right? So it debuted in the 1990 with Waters Millennium, which eventually became Empower. And now most of the major vendor actually use this format, which is much more preferred because it gives you the metadata, which is related to the sample, and afford you faster data retrieval and management. So this is the way to do and provide the necessary audit trails. Case study number 2 came from Thermo Scientific's Chromalion 7 CDS, showing the target screening of known compound using high resolution mass spec. So this Chromalion actually was capable to get data from some of the high resolution mass spec, such as the Q executive high resolution mass spec, which is pretty impressive, and also DAD diarrhea detector, right? So shown here is the ability to automatically annotate and get some of the data, but not all the functionality of the executive OB trap. The next one is an example case study of the Peak Explorer view function on the Agilent's OpenLabs CDS. For instance, if you have a whole batch of sample containing, it's very difficult to look through all of these and look for trends. And this Peak Explorer's view let you have a quick glimpse by transferring the peak data into dots, and to let you look at the whole batch quickly and flag things which are our specification are problematic samples. This is a futuristic slide, a figure, kind of to demonstrate the future, a revision of lab of the future, right? So shown here the integration with all these different pieces, such as electronic notebook, CDS, LIMS, and data management system to allow you to have the vision of a paperless laboratory where everything can be done electronically with automatic transfer of data, right? So from data generation, recording, archival, report, into archival and the automatic generation of CFA. Now, most labs are not quite there yet, unfortunately. It's easily said than done. Most company, even some major companies, are still using a kind of a hybrid system, where some paper system plus some of the electronic data. The last table is an example of HPLC system from the four major manufacturers, which happen to be the four major manufacturer of CDS also. So starting from Agilent, Shimadzu, Thermo Scientific to Waters, with their flagship CDS for their chromatographic system, which is Open Lab. ChemStation is being phased out and only retained for some special situations. And we have lab solutions for Shimadzu, Chromeleon for Thermo Scientific, and then Power, and show some of the mass spec that is more compatible. Typically, the CDS can control a single quad mass spec routinely. And also shown here are different type of HPLC, UHPLC, and the maximum pressure in different series and models. Here's my summary and conclusion, right? First segment is the perspective, introduction, glossary, requirement, characteristic, historical perspective. Second is operating principle. We mentioned about our sample testing workflow. We illustrate example from Waters and Power, show you integration, the results spectral display, and databases. Finally, some case study, also from Agilent and Thermo Fisher Scientific on CDS, and a listing of HPLC system and also their corresponding CDS. Here's my list of references, including some key references from the major CDS, and also some of the regulation and the lab the future reference. The two primary are the white papers and the chapter of a book written by Bob Masseries, and also the white paper or CDS published in 2019. Here is my final slide. We completed episode number 18, CDS, Perspective, Operating Principle, and Case Study. Thank you for your attendance, and we thanked LCGC as a channel collaborator. You're encouraged to subscribe to the channel for future videos on the HPLC fundamentals. On that note, see you next time.